Hello everyone and namaskar. This is Dr. Sudarshan Sambajirao Pedge, Assistant Professor, Department of Zoology. Kalasa Shirasika Mahavidyalaya Devani, your zoology companion, and welcome to my lecture. In the last lecture, we also studied the compound lipids and its types. So today we will learn about derived lipids and properties of lipids. So let's get started today's lecture. So derived lipids are obtained by hydrolysis of simple and compound lipids. It means these are derivatives of simple and compound lipids but do not have any ester linkage. Okay, so means that is the derived lipids, it will be formed with the help of hydrolysis of simple and compound lipids. Okay, that will be formed derived lipids also, right? So the cholesterol and ergosterol are the common derived lipids, okay, which are generally termed as sterol or steroids also. Okay, the sterol are solid alcohols which are distributed in nature. Okay, sterols are one particular solid alcohol, and that alcohol, it will be distributed in the nature also. Most of these compound lipids are complex compounds having an active hydroxyl group. Okay, most of the, if you seen in the derived lipids compounds, that time one particular hydroxyl group is present. Okay, one particular, that is an hydroxyl means OH group is present in the derived lipids also. Okay, so next, in the derived lipids we have seen these two cholesterol as well as ergosterol thoroughly. Okay, so first is the cholesterol. Cholesterol is the best known example of sterols, which is present in all animal cells and particularly abundant in a nervous tissue and bile secretion. Okay, it is an unsaturated monohydric secondary alcohol. Okay, it is an unsaturated monohydric secondary alcohol with the formula of C27H45OH. Okay, that is the formula of cholesterols. Okay, besides bile and nervous tissue, cholesterol is frequently available in cod liver oil, bone marrow, egg yolk, and wool fat. Okay, it is not present in the only in the cell membrane also. It also present in the cod liver oil, bone marrow, egg yolk, and wool fat also. Okay, the cholesterols are found in these organs. Okay, or in these body parts. Okay, another, that is the cholesterol is the crystallizes in white and waxy crystals, and it is insoluble in water but readily soluble in the fat solvents. Okay, it is insoluble in water but the readily soluble in the fat solvents. Okay, we know very well cholesterol has a number of specific uses in animal body. Few important uses I have discussed here. So about 80% of cholesterol is used in the formation of cholic acid, okay, in liver, which is turns forms a bile salts. Okay, 80% of cholesterol is formation of the cholic acid in the liver, and that alcoholic acid, it will be converted into the bile salts also. Okay, so it is also used in the synthesis of sex hormones and adrenocortical hormones by adrenals. Okay, it is also used in the sex hormone and adrenocortical hormones. Okay, so see here, the structure of cholesterol as per the formula C27H45OH. Okay, in the last slide we have seen the formula of C27H45OH. Okay, so see here, in that the one OH group is there in the last ring. You see here OH group and 27 carbon atom, and H is the 45 hydrogen atom is 45 and 1 OH. That is the one particular structure of cholesterol. Okay, so next is the, so this is the structure of cholesterol, and next is the ergosterol. So yes, ergosterol are the plant origin and is almost similar to cholesterol. Okay, if you compare the cholesterol and ergosterol, those are the derived lipid and those are very similar, but cholesterol found generally found in the animal body or animal cell, and this ergosterol found in the plant origin or found in the plant body. Okay, its formula is C23H43OH. Okay, that is the formula of ergosterol, and the cholesterol C27, right, H45OH, right, in the formula of cholesterol, right. So it is the two sterols are the substance of great physiological significance. Okay, this ergosterol and the cholesterol are a very great significance of in our body. Okay, cholesterol and ergosterol function as a precursor of bile acids, adrenocortical hormones, and certain fat soluble vitamins. Okay, that is the total is the derived lipids we also studied. Okay, in the first that is the cholesterol we have studied. After that ergosterol. After the classification of lipids, we start next topic, the properties of lipids. Okay, so first property of lipids is the odorless. Okay, we know very well pure fats are odorless, means no any kind of smell of lipids. Okay, as well as tasteless. Okay, the one spelling mistake is there, tasteless, less D झालेला आहे तिथं, yes आहे, yes पाहिजे, one spelling mistake is there is a typing mistake, spelling mistake. Okay, so first is the odorless and second is the tasteless, means the lipids are tasteless, no any kind of taste for the lipids. Okay, it means bitter, sour, sweet, no any kind of taste. Okay, it means the tasteless lipids. Okay, another the property of lipids is the colorless greasy substance. Okay, it means the one particular colorless greasy substances, and as I said in a last lecture, the pure fats are solids while the oils are liquid at room temperature. That is the next property, that is the melting point property. Okay, In the melting point property, as I said earlier in the last lecture, the as well as the pure fats are the solids, but while the oils are liquid at a room temperature. Okay, it means they are in that is the one particular. We know very well the melting point of lipids depends on its chemical nature, right? And the fatty acids with a greater degree of differences in the unsaturated and saturated fatty acids. Okay, the melting up point of, if you compare the melting point of unsaturated fatty acid as well as the saturated fatty acid, the melting point of unsaturated oleic acid is a 4 degrees Celsius, only 4 degrees Celsius, but in a saturated stearic acid is the 70 degrees Celsius. It means there is unsaturated easily melt in a only little bit amount of temperature, that is the 4 degrees Celsius, but in the saturated fatty acid, that is the stearic acid, is the very high temperature they have melted. Okay, it means the 70 degrees Celsius, that is the melting point of saturated acids. Okay, that is the one particular nature of particular chemical. Okay, so the pure fats or the lipids are insoluble in water and other aqueous substances. Okay, if you soluble in the water in pure fats, they does not soluble in that in the water. Okay, this is insoluble in water, but they are soluble in the ether, alcohol, chloroform and other organic solvents also. Okay, they have soluble but in the ether, alcohol, chloroform and other inorganic organic solvents also. Okay, that is the solubility of lipids. Okay, that is the one particular properties of lipids. Okay, another property is the enzyme hydrolysis. Okay, we know very well lipase is the enzyme, and that lipase enzyme, it will be present in the elementary tracts, or it will be present in a elementary tract of our body, right? Of any animals, our body as well as of any animals, act as these lipase enzyme act as a hydrolysis of ester bonds of triglyceride and releasing fatty acid and glycerol. Okay, that is the enzyme hydrolysis. It means if you taken any kind of lipid molecule in our body, that time our body contain one particular lipase enzyme, and that lipase enzyme, it will be hydrolyze the triglyceride and formation and release into the fatty acids and glycerol in our body. Okay, and then absorption is started. Okay, so this is the enzyme hydrolysis. Another second, the next is the hydrolysis with alkalis, that is called as saponification. Saponification means the soap formation. Okay, the hydrolysis of triglycerides with a strong bases means NaOH and KOH, means sodium hydroxide and potassium hydroxide. If you hydrolyze this in that strong bases, the triglyceride, that form or that term called as saponification. It means when the hydrolysis is complete, glycerol and the salts of fatty acid are termed or are formed, means formation of soap. Okay, one particular formation of soap. Okay, mean hydrolysis of alkalis. Okay, that time we have formation of soap. Okay, many type of industry for soap formation industry, they have used this reaction, that is the hydrolysis of hydrolysis with alkalis. uh, Triglyceride is converted into the soap. Okay, that is the one particular hydrolysis with alkalis. Another second or next is the hardening of oils drying. Okay, we know very well the air drying of oil paints, varnishes, etc. Okay, that is the air drying of oil paints. Any kind of oil paints easily air dry by the uh, hardening of oil and drying. When oxygen attacks the unsaturated oils, form hydroperoxy groups. Okay, and the next one is the animal diet. Is it used in lipids is used in animal diet? Yes, lipids are a food value, and lipids form an important source of energy and a component of the animal diet. Okay, it means it is one particular abundant source of energy with the help of lipids also. So today we have seen in this lecture, that is the um, derived lipids. Okay, in the derived lipids we have seen that is there are two derived lipids, cholesterol and ergosterol. Generally cholesterol found in all animal cells. Okay, and nervous tissue as well as bile secretion. Okay, these are the main and important functions of cholesterols we have uh, seen today, as well as ergosterol, as well as structure of cholesterol, and ergosterol is also studied in the lecture. And last is the properties of lipids is also studied. So this is it for today. Thank you very much.